Hi, this is Alessandro from the Quantum Biology Institute and the Quantum Biology DAO, and today I'm going to talk about the one-of-a-kind microscope that we're building that is going to be able to look at quantum signatures inside of living cells. So, first of all, a little bit about the Quantum Biology Institute, which is a non-profit that studies the interplay between tiny magnetic fields and biological processes, and we do believe that this interaction is mediated by purely quantum effects, namely spin superpositions, and basically this is exactly why we're building this new one-of-a-kind microscope to demonstrate that. So the first thing we did was actually demonstrate the proof of concept that tiny magnetic fields do impact life. And we demonstrated that in tadpoles, where we removed Earth's magnetic field and we observed accelerated aging in tadpole embryos. So we know that magnetic fields do something in biology. Now we want to understand the mechanism. Let's talk about how we're going to do this. I'm here with two microscopes that are exactly what you probably think of when you hear the word microscope. But these are not what our big microscope is going to look like. And there have been many microscopes, many different kinds of microscopes that are looking some at quantum signatures, some at how biology evolves, but there is no microscope that can do what we want to achieve, which is looking at the quantum signatures inside of living cells and tying the behavior of the spins that are involved in electron spin superpositions and tying that to biological function in a single experiment. We're not reinventing the wheel here. We are basically constructing a microscope that has many different things that already exist, and we are combining them for the first time. We're starting off from what is called total internal reflection fluorescence microscopy, which is a technique that is commonly used to image biological systems in a very gentle way. So we're not doing anything harsh to the cells and the proteins, unlike some of the things that are usually used for quantum measurements that are bleaching and killing the proteins and cells very quickly. So we're taking this basic biological microscopy technique and we're adding some coils to have magnetic field control over the sample. And also we're adding a new path of detection, which is very similar to what is called optically detected magnetic resonance. And this is what gives us the information about the quantum signature. So we can tell what the quantum state is in terms of spin superpositions via fluorescence signatures that we can detect by synchronizing everything to the nanosecond level and single photon detection. The first thing that we want to achieve with this microscope is to actually detect these quantum signatures, which are going to be oscillations in the fluorescence at very short timescales inside of living cells at room temperature, basically demonstrating that quantum states can survive for long enough inside of biological conditions in order to matter. After this first step, we want to use magnetic fields to change the quantum state and control it and see how the magnetic field changes, one, the quantum pattern of spin superpositions, but also the biological function downstream. And this will give us mechanistic insight on the interaction between magnetic fields and biological processes. So if we do understand how magnetic fields interact with biology, we can actually take this knowledge and apply it to literally every single biological process that exists. And so this could be a revolution in terms of healthcare, biomanufacturing, drug discovery, and literally anything biological you can think about. If you're interested in our mission and in our quantum microscope, keep following us, because in the next video we're gonna go into who is involved in the project, the different tasks that each one of us has, and basically the master plan in order to get to significant results as fast as possible.